so it's a pleasure to have with us today Dagmar Iber. She studied math and biochemistry, in fact, in Cambridge, Oxford, and before in Regensburg. And she holds both PhDs from both disciplines. Well, I would say the field, no? Correct me if I'm wrong. In both math and mathematics and biochemistry. Dagmar was a lecturer in applied maths at Imperial College London, and then moved to ETH Zurich in 2008. And since then, she has been working on, well, since the beginning, she has been working on database predictive models for understanding temporal dynamics in signaling networks. And she works really in between all thoroughly between lab experiments to the development of mathematical models and their analysis. So without further delay, it's a pleasure to have Dagmar with us, and she's going to give us the talk about computational models and fundamental principles of epithelial morphogenesis. Please, the floor is yours.
Thank you. So first of all, thanks for having me at this colloquium. And today I want to indeed talk about epithelial morphogenesis. So my group takes an interest in many different patterning systems, and we have worked on many different ones over the years. And so I had to choose one to talk about today. Oh, let me just hope the screen is not frozen. Point of view, it provides an interesting question of how you get symmetry breaks in a reliable fashion. And I will explain this a little bit more as we go along. But as you can see, the lung and the kidney, they're both branched, but yet they're looking different. And so what I will focus on is how would you determine these thousands of branch points in a reliable fashion? And also, how would you establish the tube structure? So that's yet another symmetry break because you have a bias in the outwards. And so in both cases in development, you somehow have to break the symmetry. And the question is, what's the mechanism behind it? And that's why we're interested in the lung and the kidneys. They're obviously also very important organs, but that's the motivation. And in part 2, I will dive in more deeply into the epithelial structure, and I will discuss fundamental organizational laws of how these cells organize and what's the physics behind the organization. And we got there because we were doing microscopy and we were seeing these structures. We were wondering what's the reason for them. And they had been discovered almost 100 years ago, and there were lots of models out there, but none of them could really explain it. So that was really a nice mathematical physical challenge for us to work on. And lately, we have translated that into a 3D cell-based simulation framework for these cells that have much higher geometric resolutions than current vertex models. And we hope and expect that they may prove useful to better understand epithelial dynamics in development and disease. So what shapes an organ? That's the question that we are asking when we are looking at lung and kidney development. So as I said, both organs are heavily branched, but yet it's easy to tell them apart even if you're not a biologist. And the question is, how do you establish these branching architectures? And if you're looking at mice from the same genetic background, they're actually looking identical. And that was actually the finding that got me into the field. In 2008, there was a phenomenal Nature paper by the Krasnow group. And what they found after looking at mouse lungs for 7 years, so that was one PhD student, he realized they all looked the same. And that was an amazing finding because previously people would have thought it's kind of a stochastic process. The tubes just find their way wherever they have space, but clearly not. There's some genetic mechanism behind it because if you're taking mice from different genetic backgrounds, you're finding small differences. And so the question then is, how are you establishing that branching program? And also what they realized when they looked at the lungs is that there's only 3, in fact, 4 ways of making a branch. You can either branch to the sides, then you're getting domain branching. You can bifurcate in the same plane, or you can bifurcate and then have a second bifurcation in an orthogonal plane. And as you can see, as the lung is growing, the different branching modes are used in different places, and you only have certain sequences in which they occur. And this sequence is stereotypic. And so the question is, what's the mechanism behind that? And what the biologists had already found is that you're only needing 2 pathways, fibroblastic growth factor 10 and sonic hedgehog, to make a lung. All other pathways, if you knock them out, they will modulate the branching pattern that you're observing, but you're still getting branching morphogenesis. But if you remove these 2 pathways, either of them, you're not getting branching. You're still getting outgrowth of the branching, but no new branches can form. And vice versa, if you take FGF10 or put it on a bead, put it next to the epithelium here in green, a new branch gonna grow out next to the bead. And from that, the biologists concluded that FGF10 is both necessary and sufficient to induce a branch, and that it's really the growth factor that is controlling it. So from that, we know lung branching requires FGF10 and sonic hedgehog. The other thing that biologists found out is that these patterns can emerge even if you have uniform initial conditions. Means if you have uniform expression of these growth factors, you're still getting branching morphogenesis. And if you lower FGF10 expression, then the distance between branch points increases. And all these features, they're reminiscent of a Turing mechanism. And so what we realized is that you could formulate a Turing mechanism that would give you that kind of branching. And what was different from Turing mechanisms that people had formulated before is that we realized that you could get Turing patterns with a single morphogen, with a single ligand. If that ligand binds to its receptor and upregulates the abundance of the receptor. And this you indeed find for both sonic hedgehog and FGF10. But then we wanted to know whether that mechanism could actually explain biology. And the only way to find out is to compare quantitatively to data. And the first data set that we obtained, or that was from a French group that had just published these. And what you're seeing here is an epithelial bud, which is surrounded by mesenchyme. And you see that over time, which comes from different embryos, but it's so stereotypic that we could just combine that series. And what we then did was that we calculated the displacement field between stages, consecutive stages, and then we extracted the growth field as the vector field that is outward pointing. And we then simulated our Turing model and competing mechanisms on those surfaces, and then compared the predicted signaling field to that growth field here as vector. And here you see an example of a perfect match. And then we calculated the difference and compared the mechanisms. And when you do that, you find out that the Turing mechanism can be matched very easily to those patterns, while the main competing mechanism at the time was a geometry-based patterning mechanism. And that geometry-based patterning mechanism said, like, you're not starting from a plane or whatever. You're already having some geometry and the mesenchyme around it. And so there may be geometry effects. So for instance, here, if you're looking at this, disk, if you're expressing a morphogen in the disk, but not outside, then you're going to have the highest concentration in the center. And if that concentration is now inducing growth, if the growth rate is proportional to the intensity of the signal, you would create such a bud. But if you now look at that quantitatively, what you can see is that the pattern that you can obtain, the best pattern you can obtain by looking at a large parameter range, you only have a single parameter in this model because you only have the diffusion coefficient. Then you see that the signal concentrates in a similar part as a growth field, but the match is not perfect. And so that's the best match we can get. And from that, you can see why biologists had this idea. It's not such a bad idea, but if you quantitatively compare the shape and the model predictions, it's just not as good as if you're using the Turing model. And so from that, we can say, yes, the geometry effect will certainly exist. It's also incorporated in our Turing models indirectly, but it's not the driver of this. So it must be some other symmetry breaking mechanism, and we think it's a Turing mechanism. So then we tested that in more detail. So in the lung, this ligand, the FGF10, is expressed in the surrounding structure, the so-called mesenchyme, and the receptor is here on that epithelial tube. And when you screen for parameters, we're getting a perfect match, as I just said. Now, oops. If you invert the distribution, so the receptor is now in the mesenchyme, the ligand is produced in the epithelium, you wouldn't get a match. And this kind of distribution you have for sonic hedgehog. Sonic hedgehog is an inhibitor of branching, while FGF10 is an activator of branching. If you take that into account, it works again, and sonic hedgehog can also guide branching morphogenesis. The same applies in later states. So even though the geometries are more complex, you're still getting a perfect match with these two mechanisms. Now, one thing about Turing mechanisms is that you can get different patterns for the same parameter set. So the initial conditions have quite a huge impact. So if we use noisy initial conditions, you could either get a but or you could fail to get a but. And that obviously would not be acceptable in biology. And so then the question would be, how does nature avoid this? And the solution is a pre-patterning process. So as I said, the FGF10, the ligand, is produced in the mesenchyme. It has to diffuse to the epithelium where you have the receptor. And that diffusion process is already taking care of any noise, because the geometry effects that I discussed before will be dominating, and therefore set the initial conditions, even if you have variations in the expression of the FGF10 by different cells. And if you take this into account and solve this either only on the epithelium, so if you express both ligand and receptor in the epithelium, you get many different patterns with the same parameter set. While if you separate them between epithelium and mesenchyme, you only get a single pattern per parameter set, and therefore you get the differential robustness that one observes. Now, interesting enough, biologists have done the experiment, not for us, they just did this. They expressed the ligand in the epithelium, and when you do that, then for the first time, you're getting a litter where each kidney in that case looks different. You know, we have a very similar mechanism in the kidney. And so that's in line with what we would expect. And so we would say the separation of ligand and receptor production into two different tissues is in place in order to deal with noise effects. And so finally, all the data that I showed you so far came from different embryos. The referees at the time asked us to get dynamic data. For us, that was a huge challenge because we were a computational group, we had no lab. And so that's the first experiment we ever did in the lab of a colleague. And we segmented that, and when you do that, only a Turing mechanism can predict the points of outgrowth. So again, you see the highest signaling is predicted at the points of outgrowth, while all non-Turing mechanisms cannot match the growth field. And so that's telling us that the Turing mechanism is a very good candidate mechanism for the lung. And interesting enough, we have a huge number of branched organs in our bodies. And all of these organs rely on FGF10 and sonic hedgehog to guide branching morphogenesis. The only exception to that is the kidney, and the kidney relies on a protein called GDNF and Wind. Now, both are very different pathways from FGF10 and sonic hedgehog, one would say, even though GDNF converges on the same transcription factors. So there seems to be some similarity, even though GDNF is formally a member of the TGF-beta family. So now what we realized when we looked at the regulatory dynamics of the regulatory logic of GNF and FGF10, they have exactly the same architecture. So GNF in the kidney is also expressed in the mesenchyme, the surrounding tissue, much as FGF10 is expressed in the mesenchyme, while the receptor RET is expressed in the epithelium, and binding of ligand and receptor results in the upregulation of the receptor. And so this module, which we had identified as Turing module, exists in both organs. And so we tested whether the same mathematical mechanism could also explain branching morphogenesis in the kidney. So we obtained cultures of the urothelial bud. We obtained the growth fields in the same way. And then we can compare the growth fields to the predicted signaling fields. And you can see that the Turing mechanism predicts those growth fields very well. And here in this place, initially, you didn't see any Turing mechanism, even though it is curved initially. And so that here you can see there's no signaling from the Turing mechanism. And only once it starts growing out, you see the Turing mechanism marking it. So we're not just marking curved boundaries, but rather really the spots where you see outgrowth. And we can also solve the free boundary problem. Means we have a boundary, we solve the Turing mechanism on this boundary, and now we have a normal vector field for the growth field. The strength of growth is proportional to the strength of the signal, and you can see we get very similar branching morphogenesis. We have also tested this in a couple of mutants that have very different branching patterns, but the mechanism still works. Now, different from the lung, in the kidney, we have a positive feedback between GNF and Wnt. So we have a negative feedback in the lung, but a positive feedback in the kidney. And so Dennis Menschikow, he wanted to know what purpose that could have. So he did a simulation where he either included that feedback or not. What he predicted from the model was that this positive feedback is important for the kidney to be able to come closer in the branching. And it's important in the kidney because the kidney has almost 90-degree angles, while the lung has more 60-degree angles. And so you can see that here. If I branch at 90 degrees, twice, then these buds gonna start to grow into each other. And by having a positive feedback on the ligand, they can replenish the ligand, otherwise they're gonna compete over them. And so the prediction was that if you knock out Wnt11, these branches gonna stay further apart. And that's indeed what you find. We also have done statistical analysis on that, but you can effectively see it by eye, that if you knock out Wnt11, the branches stay further apart at the same stage. And so to sum up on this first part, we can exclude a pure geometry effect in the control of branching morphogenesis in the lung. A Turing mechanism gives a very good match to the experimental data, but alternatively, especially Celeste Nelson proposed various mechanical effects. One of them coming from smooth muscles. The smooth muscle idea has not had any support from genetics, so you can remove smooth muscles genetically, and branching morphogenesis is not affected. A second mechanical idea is differential growth. So if you have two layers and they grow at different speeds, you can also get a mechanical instability that results in buds. And these buds would also have a wavelength. So you would have a characteristic distance as you do for drawing patterns. And the differential growth between epithelium and mesenchyme is certainly not required, because you can remove the mesenchyme, you can grow the epithelium in extracellular matrix, and you're still getting branching morphogenesis. However, this matrix that you have around it, the extracellular matrix, obviously you would have differential growth, or you could have differential growth between the epithelium and the extracellular matrix. And this is something we cannot distinguish easily, because the extracellular matrix and the signaling mechanisms, they are really linked. So for instance, the extracellular matrix is thinner at the tips of the bud because FGF10 is upregulating metalloproteases that are turning over the extracellular matrix. So we have here mechanochemical coupling And we will need other experiments, more experiments, in order to distinguish whether a signaling-based Turing mechanism or some kind of mechanical instability is at play. But the Turing mechanism we have tested in more depth, more detail. So here we have the lungs at different somatite stages. Somatite stages about 2 hours apart at this stage. And we can segment these lungs, and we can then simulate the Turing mechanism on those shapes. And you can see that the Turing mechanism predicts new branches many stages before the bud really emerges. Means like it's not a geometry effect, it's really placing it at the right spot. Now, when we simulate the Turing mechanism on a growing domain, you're still getting quite nice results, but you can see that there's important differences. One important difference is this asymmetry. We could introduce that. It has something to do with the dorsal-ventral polarity that is inherited by the lung from very early on. That's coming from the foregut, from the really early stages of development. The other aspect, however, is if you're looking at the branches that we are simulating and compare that to the branches that you're seeing here in the embryo, the branches look much fatter. And the reason for that is that in the embryo, you have biased outgrowth. And that's yet another symmetry break. So we just discussed the first symmetry break of placing the points of outgrowth. But once I've placed those points of outgrowth, I must have a second symmetry break that biases the outgrowth. And we were not the first to note that effect. Probably ever since people have looked at developing lungs, they have noticed that. You can see this by eye, you don't need quantifications for that, that you have more lengthening of the tubes and widening. And you see this both in 3D, that's effectively 3D images here from the light sheet, and in 2D cultures. And there was a really nice science paper by Gail Martin's group in 2011, and they quantified the left bronchus and found that the left bronchus is elongating twofold more than it widens. And they also showed that the cells predominantly divide in direction of outgrowth. And also the cell shape is biased in direction of outgrowth. And that triggers a question, what creates that bias? Why are the cells biased in that direction? And why is the outgrowth biased in that direction? And how do you get the symmetry break? Again, thousands and thousands of times during lung branching morphogenesis. And so we use the cell-based model to understand this bias. We use the vertex model, and we can parameterize the vertex model by the measured neighbor distributions and area distributions. I'm going to come to that later. It's going to make more sense later on, but let's just say we can parameterize those models based on data. So all the parameters are based on data. We don't have free parameters. Then we apply a force at the top, and we are pulling on the tissue. And when we do that, we can get a bias in outgrowth. And a twofold bias in outgrowth corresponds to a certain force that we apply. And when we use that force, we can reproduce exactly the bias in cell mitosis, in cell shape that is observed. You might just say, well, okay, you can just adjust your parameters, but you don't have free parameters. And that comes into play when you do the opposite. Instead of pulling on the tissue, we can also constrict the tissue. When we simulate that, this time with a different software, we used our own software, LBID cell, you can still get that bias in outgrowth, but you no longer get this bias in cell shape. So it means cell division, so that's the measurement here in light blue, and these are the simulations. We cannot get to that bias in cell division. So we have the bias in outgrowth, not in cell division, and there's no free parameters that we could adjust. And so based on our simulations, we would expect there's a pulling force and not a constricting force. So what could be that pulling force? So the first idea one might have is that the Turing mechanism suggests that FGF signaling is concentrated at the tips, and that this creates a bias of whatever description. And indeed, also PERK, which is a downstream signaling factor of FGF10, is clearly concentrated at the tips. However, if you inhibit FGF signaling with an SU5403 inhibitor, you affect outgrowth, you affect branching, but you're not affecting the bias of outgrowth. So both the controls and those SU cultures, they have the same bias in outgrowth. And that means that this signaling of FGF10 at the tips seems not to have anything to do with the bias in outgrowth. The next thing we tried was we removed the mesenchyme. So the mesenchyme that surrounds the epithelium, we put the epithelium in matrigel, and we measured the outgrowth both in the lung and in the kidney. In both cases, we're still seeing the same bias in outgrowth. And so that means also the mesenchyme that is surrounding the epithelium is not required for that bias. The next thing that one could think of is what I told you before, is the extracellular matrix is thinner at the tip. And that obviously would be less of a mechanical barrier. And so that might encourage biased outgrowth. So we perturbed that by using an inhibitor of the metalloproteases that turn over the ECM. This affects outgrowth and branching, as people had described before, but the bias in outgrowth is not affected. I should maybe explain this. If the points would be on the orange line, we would have uniform outgrowth. And what you can see here, we have over time more lengthening. So the length of the branches increases and the width, however, decreases rather. So we're not getting as much widening as lengthening. So we have clearly the bias in outgrowth. And we also looked at actin staining, and we see no evidence for actin at the front. And so that would not support any lamellipodia or whatever. Now, what Harold Gomez realized when he looked at the light sheet images is that those tubes are very narrow. So only at the butts do you see openings, but the tubes themselves are so narrow that with the fluorescent signal, we can't see any open spaces. And the same holds also when we do live imaging of branching lungs. And so that means it's not a clearing or whatever artifact. And people had shown before that there is fluid inside the lungs. And in later stages, there's also fluid movement. They've always accounted that to smooth muscles. And so we set out to see whether there's any fluid movement already in the very early stages of lung development when we see the biased outgrowth. And that's an experiment that Lisa Conrad did in the group. So she injected beads into the developing lung, and then she quantified the speed with which the beads are moving along. And she obtained this average velocity, average speed. And with that information, we can calculate the shear stress that this fluid flow is causing on the walls. We also know the viscosity. Someone else had measured that as 10 times higher than for water. And Steve then took the left bronchus that has been studied by everyone so far and simulated fluid flow at that velocity through that piece of through that bronchus, and he observed shear stress levels at the level that cells can sense. So that's shown here. The shear stress level obviously depends on the cross-sectional diameter of the lumen. As I said before, we can't quantitate this accurately with fluorescent microscopy, but we can be certain it's less than 20 micrometer. It's probably just a few micrometers. And for this entire range, we're getting shear stress levels that are within the range that cells can sense. And quite nicely, consistently, there is a mutant KRAS G12D, which doesn't have a cilium and which doesn't show biased outgrowth of lung tubes. And so we would conclude that fluid flow inside the lung is causing shear stress, which the cells are sensing, and translate that into a biased cell shape, which then leads to biased cell division, which then leads to biased outgrowth. So to sum up on this part, there is anisotropic growth in lungs and kidneys in early development. Based on the cell-based modeling, we can conclude there must be a pulling force rather than a constricting force. We can rule out, based on experiments, we can rule out that FGF10 signaling, or FGF signaling, lamellipodia, mesenchyme, or the ECM would be the factors. And we have evidence for fluid flow that can cause shear stress, which would explain the bias in outgrowth. Right, so in the second part, I want to now dive into the details of the epithelial architecture. So here you're seeing a lung at a higher resolution, and as you can see, that light sheet microscopy allows us to see single cells in those epithelial structures. And once we could see them, we wondered, why are they shaped the way they are shaped? Because if you're looking at a textbook or a review, then epithelial cells are typically depicted as prisms. So a prism has an n-sided polygon on both its sides, and this n-sided polygon is the same on the apical and basal side. Now, if your epithelium, if an epithelium is curved, if you have a spherical epithelium, then those cells are typically depicted as frustum or prismoid, where you have the same number of neighbors on the apical and basal side, but because you're on this curved surface, your outer side is larger than the inner side, and so you have a scaling. So that you have a uniform scaling of your polygon. Now, if you're on a tube, then the principal curvatures change differently. Means like along the longitudinal axis, you don't have any change in curvature. But as you go to the inside, this is changing. And so now, if you're looking at a polygon on the outside, you imagine what it looks like on the inside, then the longitudinal diameter will not change as you're going to the inside. But the orthogonal axis will shrink as you go to the inside. And that means that you cannot accommodate, or you can't use these frustums in order to cover a tube. So you will have to have a neighbor change along the axis, either at the surface, at one of the surface, in which case it's a prismatoid, or you have that neighbor change somewhere along the axis. And then this structure is now called scutoid, that was introduced like 2-3 years ago. And so here in this example, at this top here, you would have 6 neighbors, and then you go to a structure where the cell only has 5 neighbors. So now when we quantify the cells in our tissue, and so we follow each cell and we check how many neighbors does that cell have and how many times does it change its neighbors along its axis. Then we're not observing one neighbor change as you would have here for the scutoid, but up to 14. So that means the single cell is changing its neighbors 14 times as you go from the apical to the basal side. And if you're on this tip structure, which is more spherical, you have like 8 or 9 transitions. And when we quantify them, they're uniformly distributed along the apical basal axis. And they seem to be stochastically distributed, so follow a Poisson distribution. Now, the structure that we are seeing in our cells is more reminiscent of the rock formation in Punakaiki. So we would propose to call that geometry punakoid to go with that rock formation. And the consequence of that, of a large number of neighbor changes, is that when you look at a cell from the surface, then it appears, then it has many more neighbors than what you would think from the apical view. So here the blue cells are all neighbors of the yellow cell. You can see this one is several cell diameters away if you're looking from the surface. And that might have profound implications for epithelial signaling. But we're interested why this happens. And so for the scutoids, the idea why you would have that neighbor change in a scutoid was that of a curvature-induced change, as I just described. And so we extended that theory a little bit to really look at those many neighbor changes that we observe, and we asked, can that curvature idea, can that really explain the 14 or whatever neighbor changes that we observe? So as I explained before, as you're going to the inside, you're gonna change the aspect ratio. And we have the theory in the supplement of that paper. I'm not gonna show you the exact theory. Maybe that would have been the right place here for the ESTMB, but I'm gonna skip that, but just give you the intuition. As you're going down, as you're going to the inside, as I said, the aspect ratio changes. And what we then asked was, how much of a curvature change do you need to trigger a neighbor change, depending on whether the cell has a small number of neighbors, like 4 neighbors, or whether at that place it has already 10 neighbors. And what the theory predicts is that if it was really the curvature that would trigger it, then you should see many more neighbor changes. It should be much easier to trigger a neighbor change. So you need much less of a curvature change to trigger a neighbor change when you have many neighbors compared as when you have few neighbors. And the reason for that is that when you have many neighbors, you're more round, you're more circular. And so when you have that change in aspect ratio, it basically triggers a neighbor change fast in order to deal with that violation of what this polygon really would like to have. But when we analyze the data, we're not seeing any such dependency on the number of neighbors that the cell has. And so that means curvature cannot be the main driver of those neighbor changes. What then? And so we dived in and looked at our data more carefully. And the first thing, and that had been observed for many times, for decades, there is a couple of phenomenological laws that you observe on the apical surface. And we then asked, do we see this also along the apical basal axis? And the first one is Euler's formula. So cells on the apical surface have on average 6 neighbors. That just follows from topology. You have a topological constraint when you have a polygonal lattice. And so if you have 3 vertices, 3 edges per vertex, you expect to have 6 neighbors on average in an infinitely large polygonal lattice. And you see something close to it also along the apical basal axis. So we understand the origin of that observation. But when you look locally, you do not have 6 neighbors, but locally a different law applies. And this law is called Above-Weer's law. I didn't print it here. And this Above-Weer's law relates the number of neighbors that the cells have in the center to the average number of neighbors that these neighbors have. And instead of being 6, you get this relationship here. And as you can see, our epithelium completely follows Above-Weer's law. You see this for all epithelia that have been studied to date. They all follow that phenomenological law, but so far no one knew why. As another phenomenological law is Lewis' law, which relates the average area of a cell to the number of neighbors. And the original Lewis law has a linear relationship, or postulates a linear relationship, which we find for the basal cells. And we find a quadratic relationship, we're going to come to that, for the apical side. So where does this Lewis law and Above-Weer law come from? Let's first look at Lewis law. So we are in this polygonal lattice, and let's assume we have equal cell areas for all our polygons. If you have equal cell areas, then each polygon type would have a different side length. If you're now trying to fit that into a contiguous lattice, then we would have to deform our cells so that they can fit in. If I deform my cells, then I increase the perimeter. And so that would be, I would increase the perimeter. And if I want to minimize the overall perimeter, to minimize the contact surface energy, then this would be a problem. Now, if I follow Lewis law, then the edge length becomes very similar. And that means I can have more regular polygons in my lattice, which would have a lower combined perimeter and therefore a lower contact surface energy. Now, if I have if I follow a quadratic relationship, then all side lengths could be equal. And so I would get even more regular polygons. But as one can see here, in order to get this quadratic relationship, I would need higher variability of my areas. And so we can test that. So the prediction would then be that we would switch to an apical, a quadratic relationship at higher variabilities of those cross-sectional areas. And if we increase that in a mutant, this is now the Drosophila wing disc, if we increase the variability shown on the left-hand side, then indeed we follow the quadratic law. If we come back to the 3D data, we can see here that the apical layers that follow the quadratic relationship indeed have a larger area CV, standard deviation over mean. And they also, we can also from our theory derive a relationship between what is a fraction of cells with 6 neighbors hexagon frequency versus area CV, and you can see that again all layers follow that perfectly. And so that would mean that that Lewis law emerges because it minimizes the deviation in side lengths and therefore allows these polygons that all have different areas to fit into a contiguous lattice by being the most regular that you can be in such a setting. So what about Above-Weyer's law? Here you now see that the formula that I hadn't printed before, so 5n plus 8. So the second constraint that you have in a contiguous lattice is that your angles at each vertex have to add up to 360 degrees. Now, if you're trying to have really regular polygons that are adjacent to each other, you would not manage in most cases to add up to 360 degrees. And so you will get some deviatory angle here. And it's very difficult to derive that for like any arbitrary area distribution. But what we can do is we can take the above-Weyer relationship that we observe in the Drosophila wing disc here shown. And the white line is the Above-Weyer's law, and you see it's pretty close to the measurements. But now we can optimize this one angle here such that we're getting closest to the measurements. And if we do that, we are predicting the range of angle deviations that we should observe in the wing disc. So for a cell with 4 neighbors, you would expect a 90-degree angle. But if you're looking at all the cells that have 4 neighbors, you're getting quite a wide area distribution, angle distribution. So up to almost 140 degrees here and maybe 50 or whatever degrees at the lower end. And these ranges follow what you expect in terms of that deviatory angle that you have to allow for to enable the packing of those most regular polygons. And so to sum up on this part, so if you had polygonal lattices with the same, where all polygons have the same area, then you would expect hexagonal packing because that would give you the smallest combined perimeter. But if you increase variability, then you're gonna get the smallest combined perimeter if the cells follow Lewis law. If you further increase that area variability, then you're getting equal side lengths only if you're following this quadratic law. Now, on the other hand, if you have these particular regular lattices where the areas and the relative areas of the cells are in the appropriate way, then you can get these perfect regular lattices and they are not following Above-Weyer's law. However, if you have an epithelium with these mixed areas that are randomly distributed, then you're going to get Above-Weyer's law to get the with the most regular lattice that is minimizing the energy. And when I say energy, I mean the for the given cross-sectional areas that you have in each section here, minimize the perimeter. And that would go from apical to basal in each slice you would have an area distribution. Where this area distribution comes from, we are not discussing. So the cells have a volume distribution along their apical basal axis, which we say is given, a set. But for that set volume distribution, you're going to get the smallest contact surface energy, the smallest combined perimeter, if cells follow Above-Weer's law and Lewis' law. So it's basically, given the area distributions in each plane, you minimize the perimeter. And so this minimization of the perimeter, the effect of the energy gain that you're gaining from there is not high enough to affect the volume distribution. So the volume distribution is independent of the effect that we're discussing. And so if we now want to have, if you want to explain neighbor changes along the apical to basal axis, then we would predict that unlike what you're seeing here in the cartoon, the cells are not straight, but the cross-sectional areas would change along the apical basal axis. So we analyzed that, and indeed, you're getting large variations in the cross-sectional area. And whenever between two slices, the cross-sectional area changes by at least 10 micrometers squared, you're almost certain that you're going to get a neighbor change. For smaller changes, there's a certain likelihood that you observe a neighbor change. So where does this area change come from? It comes from the nuclei. So inside the cells, you have nuclei, and they are having a certain stiffness. And in our tissue, the nuclei take about 50% of the cell volume. And if you think about it, given the cell height, it's impossible to have, if you have spherical nuclei, they would really stick out. So you can see here in those pictures, the nuclei are not spherical, they're already like squeezed together, but yet wherever you have the nuclei, they widen the cells. You know, it's basically a mechanical interaction between the cells and the nuclei. You can see this here, you have an almost perfect correlation between the diameter of the nucleus and the cell. And that means wherever you don't have a nucleus, the cell is much thinner. And you see this here, you get a neighbor change wherever the nuclear cross-sectional area also changes. And you're seeing most neighbor changes at the edges of the nucleus. And that's what you're seeing here, wherever you have that rapid change in cross-sectional area. And now the nuclei are moving from the apical to the basal side. So you have cell division and mitosis state. The nuclei are at the apical side, and after that, they're going down to the basal side. And then for the next cell division, they're coming up again. And so we predicted that you would see neighbor changes when you look over time. So we cultured our lungs and kidneys in the light sheet over time, and then we can segment the cells over time. And when we do that, we can only follow few cells, so you only get imperfect correlations. But the larger the cross-sectional area, the more neighbors the cell has, and we can see this really over time, how those neighbor changes are happening in this patch. So here we are focusing on the orange cell, and we can see that over time, we are getting constantly about 8 neighbor transitions. So it's happening all the time because the nucleus is pushing through that tissue. And so to wrap up this part here, using light sheet microscopy, we can now see the 3D organization of cells in epithelia, and we can, after almost 100 years, we can now understand the origin of Lewis' law. Lewis' law ensures that if you have a polygonal lattice of mixed cross-sectional areas, then Lewis' law, if the cells follow Lewis' law, you have the most similar side lengths of those polygons, and that means you have the most regular polygons. Thanks to Above-Weer's law, if you follow Above-Weer's law, then you effectively minimize this divisory angle that you need to have in order to fit into that contiguous lattice, again, having the most regular polygons possible. And these two laws apply along the entire apical basal axis because the system is in mechanical equilibrium. So you have to have a balance of forces no matter where you slice. That's why we could slice everywhere effectively and always observe Lewis' law and Above-Weer's law. Now, as I said, we use that insight to develop a 3D cell-based tissue model. So different from a vertex model, we have the cells are deformable, so they're completely meshed and they're completely deformable. They organize according to physical laws. So we have cortical tension, we have a membrane elasticity, we use an inner cell pressure to control the volume, we have a bending elasticity, and we have intercellular forces to model adhesion. The cells are polarized, so we can have different properties on the apical and basal side. And here you see an example of like how deformable the cells are. And we can obviously also have cell division, so we can divide them any way we want. And the thing I'm most excited about at the moment is that we can also import the geometries that we have extracted from our light sheet images. And so you're seeing here the tissue that we obtained from light sheet microscopy, the epithelial cells and the nucleus. We can import that into the software, and then we can optimize our parameters such that we maintain that shape. And in that way, we can now start to understand what determines the cell shape. Because what I told you so far was what determines the neighbor interactions. That means what determines the polygonal shape in each cross-section. But now we can understand what determines the volume distribution along the apical basal axis. And with that means we understand what are the physical principles of epithelial organization. And with that, I want to close. In the first part of my talk, I told you about Turing mechanisms and how they can explain branch point selection in lung and kidney branching morphogenesis. I talked about fluid flow and how it can guide the elongation bias that we observe in early lung and kidney development. And in the last part, I was diving into the epithelial structure of these organs, and I described the physical principles of the 3D organization and showed you our latest software for 3D simulations. And that is one snapshot of my group. It's already outdated again. It's always difficult to keep up. So many thanks to them for doing this nice work, and thanks to you for listening.